Dear student, my name is Dr. Mohammed Irfan, and the topic of our experiment today is to assess digestion of starch by salivary amylase enzyme. By the end of the practical, students should be able to 1. Define carbohydrates, enzymes, and digestion. 2. List types of carbohydrate and give their examples. 3. Describe saliva with respect to secreting glands, composition, function, and stimulating factors, and describe the procedure of starch digestion by the enzyme salivary, alpha, amylase. Requirements for the experiment are test tubes with rack, saliva/alpha amylase solution, starch solution, potassium iodine solution, burner/lighter or water bath at 100 C, Benedict's reagent, alpha amylase solution, and starch solution. Carbohydrates or organic compounds consist of carbon, hydrogen, and oxygen. They are the main source of energy for the body in the form of ADP. Different forms of carbohydrate are monosaccharides, for example, glucose, galactose, and fructose; disaccharides, for example, sucrose, lactose, and maltose; and polysaccharides, for example, starch and glycogen. Nutrients can be absorbed and used as a source of energy only after being broken down into their monomers or the simplest form. This process of breakdown of substances into their simplest form is called digestion.
Process of digestion can be divided into two types: mechanical digestion, i.e. breakdown of food through mechanical action like process of chewing and by movement of muscles of the stomach, and chemical digestion, wherein breakdown of food take place by the action of chemical substances called enzymes. Enzymes are protein in nature that increases the rate of chemical process of digestion. Begins with the entry of food in the mouth cavity mechanically through chewing and chemically under the influence of enzyme salivary amylase. Sight, smell, and taste of the food stimulate secretion of saliva from three sets of salivary glands. They are 1. Parotid gland, that secretes mainly watery or serous secretion. 2. Sublingual gland, that secretes mainly lubricant or mucus secretion. And 3. Submandibular gland. This slide shows the location of these salivary glands. Parotid gland is situated just in front of the ear, sublingual glands are situated below the tongue, and submandibular glands are situated below the mandible bone. Saliva is composed of two parts: 1. Serous part, having the enzyme salivary amylase, which is also called ptyalin or alpha amylase. This enzyme is responsible for the initial digestion of carbohydrates, particularly the starch, and is secreted by the parotid gland. And 2. Mucus part, performing lubrication activity and mixing the food, thus helps in the friction-free movement of bolus through the digestive tract. Digestion of carbohydrates is completed at the level of brush border of small intestine by the action of enzymes leading to conversion of disaccharides into monosaccharides. For example, maltose is converted into two molecules of glucose, sucrose is converted into glucose and fructose. This slide summarizes the steps of procedure for today's experiment. We will take two test tubes and label them as A and B. Add 1 milliliter of starch solution in both the test tubes. Add the enzyme alpha amylase only in test tube A. That will digest carbohydrates. Test tube B will be without enzyme, so there is no enzyme activity expected, and starch will remain as it is. That will be confirmed by the addition of 1 or 2 drops of Lugol's iodine. Add 1 milliliter of water in both the test tubes to balance the volume. Lugol's iodine will change in color as starch is detected. So you can expect the change only in test tube B as it does not contain enzyme. To confirm the digestion of starch by amylase enzyme, in the next step, 1 milliliter of Benedict's reagent is added in both the test tubes. In the last, heat or boil both test tubes in water bath. A color change from blue to green, yellow or red is expected in test tube B, where the digestion has occurred and starch is converted in disaccharide.
We'll use an iodine solution to test for the presence of starches. Normally this solution is this yellowish color, but when this solution mixes with starch, it chemically reacts, becoming a very dark purple or black color. This makes it evident that starch is present in the solution. Our iodine solution has been used to show that both of these test tubes contain starch. To one test tube, we'll add an enzyme that will digest these starch molecules.
The enzyme we'll be using is called amylase. It's the same enzyme that's found in your saliva, and allows your body to start digesting starches as soon as you begin to chew your food.
We'll place both of our test tubes into a warm water bath. This will allow our enzyme to work much faster.
Iodine makes the starch solutions black, but once the starch has been digested by the enzymes, that black color will go away.
You can see here that after our enzyme was allowed to work, that's exactly what happened, and the color in that test tube went away.
Now that the enzyme has digested our starch, we'll test to see if it was broken apart into simple sugars. To do that, we'll use Benedict's solution. Benedict's solution tests for simple sugars.
We'll take some of our digested starch and place this into our Benedict solution. This will allow us to test for the presence of sugars. Then we'll place our solution over a flame so that it can be heated and observe whether or not a precipitate is formed. It's very important that whenever you heat the contents of a test tube, you take the proper safety precautions.
Dear students, it's the experiment. I hope you have followed it properly. Thanks a lot, and bye.